Okay, so thank you very much to everybody for the organization of this very nice workshop, especially to Veronica and the director of the center, and also to Marcela, that actually was supported from Chile, where Harvard has an office. So today we'll tell you about a project that we've been working for the past, I would say, 14 years or so, that deals with a vaccine against this pathogen, the respiratory syncytial virus. But before I'll tell you a little bit about the structure of the institute, which is a center of excellence that just focuses on translational research and goes from on areas like cancer, autoimmunity, endocrine diseases, as well as infectious diseases. So we collaborate, and some of these studies are translated into clinical studies where we hope we could eventually reach the community. That's the plan of this center. And it's based on 3 universities, Catholic University, University of Chile, Universidad, and recently a university in the north of the country. It's called Universidad Antofagasta, is joining us as a new component of the institute. So we think this flow of knowledge from basic science to clinical application. And one of the things that we do, we mostly publish papers. Recently, we were highlighted by these journals called Nature Index, that is part of Nature Press, where they highlighted 5 countries: Japan, China, Germany, Australia, and Chile, in terms of various programs that promote the collaboration between different teams. And in Chile, they highlighted 4 centers. One of those was our center. Also, this institution is called FOCIS, or FOCIS. It is the Federation of Clinical Immunology Societies. It's based here in the US, and they give this nomination of Center of Excellence to groups of scientists doing research on immunology. And we were invited to apply in 2015, and early 2016 we were appointed as a Center of Excellence. And this is the map of the world. The slide doesn't show too well, but as you see, most of the centers are in the northern hemisphere, and some few of them are in the southern hemisphere, in Australia, Brazil, and now we recently joined. And there's an Argentinian one that might actually begin soon. So most of these centers are, as I told you, in the US, Canada, and Europe. So our approach is shown here. So this is a slide that I borrow, that I modify from a book that is called Pasteur Quadrant. And this is a nice slide, it's a nice book, because it provides an approach to move from basic research to final application. And for this symposium, I think this notion is very important, because in countries like ours, Latin American countries, most of the medical applications come from research that is done elsewhere. And here I'd like to show you one example that we think have a chance to take the investment, which is mostly, at least in our countries, derived from the state, into something that benefits the people that actually are paying those taxes. Because, I mean, they don't really, they appreciate that we publish papers, but papers, we scientists like them, but people that actually pay the taxes will like to benefit from the research that we do. And I recommend this book. It's called Pasteur's Quadrant. The author is Stokes. It's a very nice book about science and technology and innovation in the US after the Second World War. So we focus on this quadrant. It's called Use-Inspired Basic Research, and it allows you to, on one hand, improve understanding, so do science based on curiosity, but at the same time, you can translate some of that knowledge into improving technologies, which is what society needs. So we do that focusing on these cells, immune cells. In my case, I'm an immunologist. We use this approach to study infections, cancer, autoimmunity, endocrine disorders, and cardiovascular diseases. These are the areas that we cover on this institute. And now, speaking of my research, I focus on the way dendritic cells capture pathogens. So this, for example, is a virus, and then they capture the pathogen, and they prime T and B cells that later go and control, hopefully, the infection. And this is an example of what we do. So we focus how the cells interact. This process is called the immunological synapse, and is required for pathogen immunity, required for tumor immunity, and sometimes fails and causes diseases like multiple sclerosis or lupus, for example. So this yin and yang between the immunity and autoimmunity is controlled by these cells. So when they prime the immune system, they induce immunity, and when they shut down the immune system, they induce tolerance. So these processes are required both at the same time. So if we want to focus now on the antiviral response, we require the activity of these cells, T cells. So this is an animation that was made by this faculty at Melbourne. I really like to show it because it describes very well what I like to, the data I'll show you today. So cytotoxic T cells are the focus of the talk. They usually, when they are confronted with a healthy cell that has self-antigens on the surface, it doesn't do anything to the cell, just is tolerated. But in contrast, if the same cytotoxic T cells sees cell that's been infected with the virus, there is a molecular process that leads to activation of the cytotoxic T cell here. So now it becomes activated, produces cytokines and other molecules, and eventually you will see that at the interface between the two cells, this T cell releases some molecules. Technical name is perforin, that actually makes a hole in the membrane of the infected cell, and another molecule known as granzyme that is able to go inside the infected cell. This is the granzyme molecule, and triggers the death of the infected cell, and by doing so is able to control the replication of the virus. So these kind of cells are the ones that you need to produce if you want to immunize against a virus. So as I told you, this process is good, it's beneficial for us because it's able to control bacterial or viral infections, but as I will show you in a minute, some of these pathogens are able to block and interfere with this process. And that's the case for the respiratory syncytial virus. So what about this virus? It's a pathogen that is, repeated the same slide, 1 second. Let me go. So this virus is everywhere. It's all over the world. In Chile it can be pretty bad. About 80% of hospitalizations in the winter are due to RSV. Just this single virus leads to a major, sometimes collapse in the public health system in this country. So, and there are no vaccines. There's one monoclonal antibody treatment. It's super expensive. It's palivizumab, which is actually effective, but does not confer long-lasting immunity and has some side effects. Anyway, so I want to show you about some basic results that we obtained when we measure the function of the T cells, those cells that I showed you were killing other cells. In normal situations, they get activated. When they are confronted with infected cells with RSV, they do not work. They just shut down. So we went ahead and tried to understand the process of inhibition and found that if you look here in yellow, these are T cells, and these are cells that are infected with the virus. In this case, with no infection, you see synapse assembly, but when there is infection with the virus, assembly of the synapse is reduced. So we can quantify that RSV was actually reducing the establishment of synapses that are required for priming T cells. So we promote the hypothesis that was a protein from the virus that was interfering with this process. And now the amazing thing is that RSV has only 10 genes. We people have about 25,000 genes. So this microbe with 10 genes is able to create all the problems that people suffer all over the world. The good thing of having only 10 genes is that we could go one by one testing which one of those molecules is the one interfering with this activation of T cells. And we did that in my lab, in collaboration with a lab in NYU, using several different systems. I'll show you one here, when we can actually add different components of the synapse to a lipid bilayer, and then different RSV proteins. And then we come with live T cells and measure the synapse assembly. And in absence of any protein, we see that the T cells create this dot here. This is the synapse. When we add a protein from RSV that is called M2, we still get the synapse and cells look like activating. But when we added the N protein, which is one of the components of RSV, there was no synapse, and the cells even suffer a major change in the shape. So this protein was doing something to the cells of the immune system. And these are movies where you see live cells. So here the control situation, each yellow dot is a synapse. Here in the middle, you have the same experiment in the presence of the RSV N protein. As you see, there is no synapse. So what we can conclude for this long basic science study is that basically, first, RSV can impair the capacity of dendritic cells to prime T cells in a contact-dependent manner. We know that RSV interferes with this establishment of immunological synapses with T cells. And the most important thing is that we were able to identify the component of RSV that was actually blocking this process. So with that knowledge, then we could think that N, the RSV component that is blocking this process of immune system activation, we could create a vaccine. Now, making a vaccine for RSV is not easy. There was a study in the 60s, a clinical study actually that here in the US that led to people, the small children that got vaccinated, to develop a more serious form of disease, as now is known as disease enhancement. And the way it actually took place is that this inflammatory response that is triggering the lungs due to the RSV infection, and which is the real cause of the airway obstruction, was enhanced by the vaccine in the 60s. So if we want to prevent RSV disease, we need to vaccinate with a formulation that promotes a different kind of immune response, one that is mediated by T cells, produces gamma interferon, and leads to IgG2A antibodies. And to do so, we created with something that I will explain in a minute, that we think is able to lead to this kind of immune response in babies, in newborns. That's the goal. So we decided to use BCG, which probably many of you know well. This is the current tuberculosis vaccine that's being used in most of the planet. And the reason for using BCGs first is safe and massive. For the use of BCG has been safe and massive in newborns all over a century against tuberculosis. So it has a very long story of safety for vaccinating very young kids. We can modify this bacterium and generate a recombinant strain expressing antigens. It has a very strong adjuvant effect, so it promotes immunity. And most importantly, if we want to induce the right immunity in newborns, BCG is an alternative for doing so. This is the use of BCG currently in the world. As you see in yellow, most of the planets uses BCG, with the exception of the US and Canada and Italy, but most of the world remains BCG user. Also, we're thinking that it could have this vaccine could have a potential use on the elderly, because BCG has been shown in elderly people to be safe as well as it is in newborns. So we did studies in mice. I'll show you just a few data that we published in several papers. So during the experiment, the control mice, they gain weight. Mice that are infected with RSV lose weight, and you can see the room is too bright, but you see here in yellow, in green, viral replication in the lungs. And these are vaccinated and infected mice, and there is protection in weight loss as well as prevention of viral replication, and we published this in PNAS in 2008. These are MRIs for 2 mice per group. So control mice have no inflammation. RSV infected mice show signs of inflammation in the lungs, and vaccinated mice and then challenged with the virus look just like the controls. So these are, as I said, several papers. This is just one example of the immune response. So this is the production of gamma interferon by T cells in response to the vaccine, all in the presence of the RSV antigen. And when we measure antibodies against RSV, in the absence of vaccine, there are very few antibodies that are considered positive. So the IgG2A, IgG1 ratio is low, it's below 1. But if the mice are immunized and then challenged, we get significant, not only an increase of the ratio, but the ratio becomes above 1, suggesting that there is more IgG2A produced in response to the vaccine. So we know exactly the mechanism. So the recombinant BCG induces very good T cells, and these T cells help B cells to produce antibodies that are eventually able to neutralize the virus. That's the way the vaccine works, and we have several papers supporting this notion. So based on these results, we needed to confirm that since we hope to replace the BCG vaccine for tuberculosis with our vaccine, that is BCG plus RSV, we needed to define and show whether we could induce an immune response against tuberculosis antigens. And this is one of the experiments we did. This molecule, PPD, is a tuberculosis antigen. This is the regular BCG that is produced by, that is actually used in people. This is the control, and this is our BCG. As you see here, 24 hours after immunization, we get equivalent results between the BCG currently being used in newborns as compared to our BCG, both at 24 and 48 hours, suggesting that our vaccine is equivalent at inducing tuberculosis immunity. So, putting this into perspective of this symposium, we've been working all this time with significant support from the government and some agencies. So we have agreements with the NIH, with CERN, with the Ministry of Health and Economy in Chile. For example, the NIH was able to support one of our GLP trials that we did here in the US. In this model, it's called cotton rats, that we have no access in Chile. Also thinking on making this vaccine accessible, we have an agreement that is in place currently between the university and the Chilean government through the Ministry of Health, so we can ensure that the global program of immunization of the government is actually able to absorb the cost of the vaccine and then make it accessible to most Chilean newborns. But this is a long way. So we, as I told you, finished the preclinical. We needed to do a phase 1, which is a safety study. Then we need to do a phase 2 that measures the immune response and defines the dose of the vaccine. And finally, phase 3, that is pretty much a vaccination campaign considering the size of our country. So we'll tell you a little bit about the progress we made here.
In order to do anything, this is a global problem. RSV is a global program, so if we wanna make a vaccine, it has to be a global vaccine. So we had no access to GMP facilities in Chile, so we decided to create a collaboration with this foundation, AERAS, which is in Rockville, East Coast, near Washington, D.C., and with their support and also funding that we were able to raise, we made about 30,000 doses of the vaccine in a GMP facility that they have, and that is actually approved by the FDA. Intellectual property is important, so we have a patent application from 2007, and also we have good papers. Papers are important to support the quality of the research that is behind the vaccine. So having the GMP material, then we can go ahead, we will think on doing a phase 1 study. Never a phase 1 study was done for a vaccine in Chile under FDA and GMP conditions. So we set up on doing that study in adults, healthy volunteers between 18 and 50 years old, and we began this study in 2017 after getting approval by the Chilean FDA equivalent, which is called Instituto de Salud Pública, the ISP. When we got this approval, was very ceremonial, because it was the first time that this institution in the history of the country was actually approving the use of a vaccine made by the Chilean science for a phase 1 study. So after getting the approval, which was in March, almost a year ago, we went ahead and started recruiting people randomly for the study. And the study was actually also enrolled. We enrolled in the clinicaltrials.gov. I think it's important to do this. It was highlighted by this institutional path. Is part of the one of the five foundations that are supported by the Melinda and Bill Gates Foundation for different major diseases, and they highlighted us here as undergoing a phase 1 study, as well a preclinical study we're doing with an antibody against RSV. So we got both antibody and cellular immunity against tuberculosis and against the RSV antigens. We're still analyzing this data. It's a double-blind study. So it looks like we have immunity, but we still don't know who actually got the vaccine and the control. But this is the way the overall picture of the study looks. In collaboration with the NIH, we are currently trying to get an IND approval. We had already a first approach with the FDA through a pre-IND, and we had a very promising, encouraging report for the vaccine according to the FDA. So again, just to remind you, this landscape of RSV vaccines. So path, initially we were here. Very few of these vaccines actually have been able to move into the clinical studies, and we are now happy to say that we completed the phase 1 and got excellent safety and immunogenicity data. So in conclusion, we think that our recombinant vaccine is able to protect against RSV. We also have another vaccine for another respiratory virus. It's called metapneumovirus, but I won't show you that data today. We know the mechanism, exactly how the vaccine works, induces T cells and antibodies. We completed the GMP production of the vaccine here in the US. We got approval for the formulation and the phase 1 by the ethical committees, the local and also the Chilean regulatory agency, and completed the phase 1 showing safety and immunogenicity. Okay, and I'm going to finish now with a little of science again, because I'm a scientist and I cannot not show you this. So one of the things that RSV does in children is inducing neurological symptoms. So about 2% of children that get RSV suffer from CNS problems that become clinical. They're serious problems. but no one had studied before whether RSV can influence learning. So we did studies in mice, and we have a study that just completed in babies, where we did learning tests. So one learning test is this one. It's called the Morris water maze. This is a pool full of water that is in a room that has some visual cues, and the pool has a platform that is hidden right at the level of the water. So the first time you set up a mouse or a rat there, it doesn't know where the platform is. So it swims randomly until eventually finds the platform and rests, and then realizes if the rat is able to learn that is near the triangle and far from the circle. So the second time you do the test, knows that has to swim away from the circle towards the triangle. Okay, when you do this test in control animals in green, first times they take about 80 seconds to find the platform. And the second time they're pretty fast, 20 seconds, and they get pretty good at it up to a few seconds. When you do the test with a rat that was exposed to RSV 6 weeks ago, so normally disease lasts for 5-7 days in the animals. So these animals are not sick. They had RSV. This is a sequela of infection. You see that the first time they do it equally as the controls, but then it takes them longer to learn how to find this platform. These are the movies. I won't show you the movies. We did a second test. This result from my student was very exciting because it was very surprising. This is a respiratory pathogen that is causing some problems in the CNS. So we did the second test. It's called marble bearing behaviors. So these animals, they live in a cage with bedding, and anything that is on top of the bedding, they hide. If there is food, they hide it from competitors. If they're pups, baby mice, they hide them from predators. And after half an hour, just a control mouse that has not been exposed to RSV, 30 minutes later is able to hide all the marbles underneath the bedding. If you do the same test in a mouse that was exposed to RSV 6 weeks, even 6 months after infection, we see this, a significant deficiency in the capacity of hiding the marbles. So these are two different tests that show the same thing, that RSV leads to learning problems. Now, when we do the test after immunization, we see that the vaccine is able to sort of revert this problem, and it's suggestive that what actually happens is that after infection of the lungs, we have data, and we've published that data, we think that the cells go back to the circulation, and some of them are able to reach the CNS and lead to learning problems, which, as you will see in this final slide, this phenomenon that we see in animals actually is taking place also in small babies. We have a study done in a hospital south of Chile with lactating babies. This study was done by by a pediatrician and a psychologist, and they showed that kids that had severe RSV don't do as well in this learning test as kids that did not have RSV or had just mild RSV. So that's what I wanted to tell you. This is my lab. We did this work 100% in Chile in collaboration with various funding agencies in Chile and international. And that's it. If you have any questions at the end, I'll be happy to answer them. So thank you very much.